So we have seen a lot of micropropagation techniques, different types of culture like callus culture, and different types of regeneration methods. So we'll just see some kind of, since we are talking only about clones means genetically identical to the parent. Because in tissue culture, we always take the somatic part of the plant and raise the whole plant. So normally, naturally, you will not expect any kind of variation to occur because the parent plant and the plantlets which are regenerated from them both belong to the same genetic makeup. Both will be diploid. There is no sexual reproduction involved in this. There is no crossing over. So normally we should not expect any variations. But there are variations which are desirable variations. They are called somaclonal variations, combining both the word somatic and the clone. It is called somaclonal variations because there is some kind of change seen in the regenerated plantlets by tissue culture. So people have made use of these changes and they have further multiplied such plantlets with desirable changes. And we have a lot of revolution happening in the agricultural area, uh, chiefly because of somaclonal variation. So many horticultural plantlets, crops have been released which have heritable somaclonal variations. That means they will be able to pass on the changes through sexual reproduction to the uh, daughter cells or the future plantlets. So heritable variations are always, desirable because if it is not heritable, then it is of no use to us or to the farmers who are going to, actually do the work. So natural reproduction methods always, they pay more, what to say, they are more featured than tissue culture. Not everybody is going to practice tissue culture. Only the scientists will practice tissue culture. But rest of the plants growth or harvest or whatever is going to be taken care of by the farmers. So whatever traits, whatever desirable characters the scientists are going to induce or infuse in the tissue cultured plants, it has to be heritably passed on to the seeds, okay, for the next harvest. Otherwise, it's of no use. So, such heritable characters which can be passed on, developed chiefly by tissue culture or by micropropagation, is called somaclonal variation. Very simply, somaclonal variation is the genetic variability which is regenerated during tissue culture or any kind of plant variants derived from any part of the plant. So genetic variations have been produced and it can be detected as phenotypic traits. Only then we'll be able to know that there is a variation occurred, because all kinds of variations are phenotypically expressed. So that is more desirable. So we have different terminologies: gametoclonal variation, somaclonal variation, protoclonal variation. So somaclonal we have seen, gametoclonal are variations which are shown by gametic culture. We have haploid culture. You will have a detailed theory aspect of it. Uh, so haploid cultures is when you culture, when the explant is a haploid tissue like anther or ovule or the ovule tissue of the ovule, the ovary. So what the egg, the actual the mega gametophyte which you see inside. So those are all haploid parts of the tissue. So when you use those part as explant or to raise callus, they form gametoclones. So they are haploid in nature. So they will have certain desirable characters which you can make use of by micropropagating such explants. So where there are variations seen even in such cultures, they will be truly haploid. So those variations are called gametoclonal variations. So when you use a protoplast, isolated protoplast to form tissues, callus tissue by fusion, by fusing protoplasts of different species or different genera, when you raise callus culture, you can see variations even in such cases where it is called protoclonal variations. So these are just 2 mark questions you might get. So how are the somaclones named? They are actually, you can say that directly, every F1 generation you get from parent, you get F1 generation in genetics, and then F1 length you get F2. Similarly, the somaclones that are generated from the tissue directly are called R0 or R. And then the next generation is R1. Then subsequent generations, you get R2, R3, R4. So that is the way they are named in subsequent generations. So important features of somaclonal variations are karyotypic variations, definitely. And there may be some isozyme characteristics. Isozyme characteristic means structurally they will be the same, but functionally they will have different functions. So for example, like isozyme. So any expression is heritable, that is genetically characterized. Any kind of expression, phenotypic expression, enzyme characteristics, any kind of expression is genotypically characterized. So you have direct karyotype changes will be there, direct changes in the genes will be seen, changes in the level of the isozymes. So most of the enzymes, you can say some of them are specific enzymes, some are isozymes. That is, they are the same structure, but they have different functions at different metabolic reactions. So you, if you have isozymes, also you can see variations. So you can't actually, structurally they'll be similar, but phenotypic expression will be different. So you can directly see karyotypic variation, that is variation in the genes or the chromosome structure, or the gene, like a mutation, for example. Similarly, in the enzyme characteristics, the morphology of the somaclones also will vary. So you have cali clones. Clones of the callus means it is termed as cali clones. Clones of the meristemna, it is called mericlone. So I've already told you about protoclone. So heritable mutation is desirable, which will persist in the plant even in the field conditions, because it should not be very vulnerable. It should not get lost when the plant moves to the field condition. So characters may be lost also because they are not passed on as stable characters. They have been, they are actually, they have somewhere during the micropropagation phase, they have been instilled into the tissue. So they might even leave the tissue. Okay, so but desirable heritable mutation will should persist in the plant population even after the plantation into the field. So variation, you have both qualitative as well as quantitative variation. That means you can see, I mean, the different structural aspect as well as the quantitative yield aspect also, you can see variations. That is, qualitative means the nature of the gene, nature of the, I mean, what to say, variations in the genetic segment of the DNA. And quantitative, how many such changes have been taking place. So for example, if you have 2 3 mutations, the number of mutations also is to be taken into consideration. Type of mutation is qualitative, number of mutations is quantitative. So when you say somaclonal variation, it is both genetic and epigenetic. Somewhere during the lecture, some time before, I have also told you about what is epigenetic. Genetic is heritable, we all know. Already it will be existing variations in the somatic cells. You can, uh, as a result of mutations, as a result of sudden changes in the structure of the DNA, these variations can enter the callus and they can be passed on to the our next generation. So these occur at high frequency. So genetic variations are very commonly seen. It could be due to any kind of factors. It could be influenced by the environmental factors. If you are giving some stress to the growing callus, it can lead to genetic variations, which is heritable. So in that condition, it might be able to produce more amount of the particular metabolite or whatever you want, the commercialization of that particular product. So in that case, it becomes heritable variation because it is caused by mutations and other genetic changes. You have another kind of variation called epigenetic variations that are non-heritable. They will be present in that batch of, you say, no, R naught or R1 or R2. In R2 you might see it, but R3 you might not see it. So epigenetic variations are generated during the culture process, tissue culture process. Temporary phenotypic changes may be produced, occur at low frequency. So that is only desirable, because if epigenetic variations occur in high frequency, then there is of no use. It cannot be commercially exploited because it will be lost before the plant goes to the field. So epigenetic variations are unpredictable variations. At particular environment, it might arise at that particular lot of cells, but when you try to clone the next generation, you might not find the variation. So they are temporary phenotypic changes, but they also are a part of somaclonal variation. So causes of somaclonal variation pathomna, you have biochemical causes, genetic causes, and physiological causes. The genetic causes, to some extent, whatever is there in the tissue, that is going to be the cause. But biochemical cause, you can say, whatever you give in the medium, whatever the media ingredients, if there is any change in the ingredient, if even for example, two different labs will give the same ingredient, but they will be different in their biochemical nature. So we say, you have so many companies. You have Nice, you have Merck, you have Fisher, you have so many scientific companies supplying sodium chloride. But sodium chloride of Fisher will be slightly different from sodium chloride of Merck. Merck will be highly pure. So when you use a small, just a single ingredient of a different company in as a medium constituent, it might lead to some variations. So they say Merck product is the most pure product, but everybody cannot afford Merck product because it is the costliest. So anybody, small level tissue culture person may use a Fisher product or Nice product. It will yield a different kind of variant than what is obtained by Merck. So biochemical causes also produce homoclonal variations. Physiological cause, definitely, because environmental factors plays a role. The amount of light entered, the temperature of the place, if there is a slight variation in any kind of callus physiology, that will definitely show it out as a variation. So these are the main causes for somaclonal variations. So when you see each one detail, you can see quantitative variations as changes in chromosome number. You can see you have already studied all these things. Aneuploidy, polyploidy, translocation, inversion, changes in chromosome number can happen during any phase of the culture process. So gain or loss of one chromosome, or gain or loss of the entire chromosome. So pieces of the chromosome may be inverted or translocated or switched over. So this is quantitative change. If you say the qualitative change, you can see the change in chromosome structure in the, as in deletion or inversion or duplication or translocation. You can see mutation of a particular gene, a particular base pair, where you can see it will involve transition, transversion, insertion, and deletion. There can be insertion of a particular nitrogen base or a deletion of a particular nitrogen base or inversion. So all these cause qualitative change. So plasma gene mutation, actually that is also involved in somaclonal variation, and transposable element activation. So transposons are jumping genes, you already know. Depending on the physiological conditions, you might expect certain classes of genes to literally jump from one place to another place in the entire chromosome. So that will also, it will exhibit some amount of phenotypic variation. Then sequences, changes can happen in the DNA sequence directly, where you can see altered fragment size. You can detect the changes in the DNA by restriction fragment, RFLP, restriction fragment length polymorphism, or you can detect the change in the protein by, all these can be detectable by seeing the DNA as such, or seeing the changes in the protein band. Or then you can also see, look for methylation of DNA. This is an important cause of this is an important, what to say, epigenetic variation. So most common cause of epigenetic variation is methylation of DNA, where the amount of DNA which is methylated, the amount of histone proteins that are methylated, they will directly cause a phenotypic change. It might not be heritable, it could be heritable also. Can't say that it will not examine, definitely it will not be heritable சொல்ல முடியாது, it could be heritable also. So these are some of the qualitative and quantitative aspects of genetic causes of somaclonal variation. When you say biochemical cause, lack of photosynthetic ability due to alteration in carbon metabolism. So any ingredient, actually when you have it as callus culture itself, you have to supply the carbon source actually. So when you want to take it to, I mean, when you go in for organogenesis, you have to allow it to photosynthesize by itself. In that case, there might be some alteration in the metabolism, which can lead to some changes in the phenotypic expression. So every plant has a common pathway for starch biosynthesis. If the callus, due to some reason, due to some physiological differences, it synthesizes using a different pathway, it might lead to some kind of variation, as they have seen that biosynthesis of starch via carotenoid pathway. So some calli, some due to some environmental condition or due to some reason, the callus uses a different kind of pathway, a pathway which is used to synthesize secondary metabolite, carotenoids, secondary metabolite compounds. So normally primary metabolites like starch are synthesized using the Calvin cycle pathway normally. But if they make use of a pathway which is used for secondary metabolite synthesis, the carotenoid pathway is a secondary metabolite pathway. So if there is a change in the pathway of synthesizing starch, there can be a variation. Similarly, common general nitrogen metabolism, if there is any change, that also can reveal a variation in the calli. Okay, so physiological cause could be many things. I told you culture conditions, any kind of environmental culture conditions will lead to somaclonal variations. And another important thing is growth regulators. So if there is, during the optimization experiments, if there is any changes in the growth regulators, the hormone, which we use as an ingredient in the medium, that they also may cause a somaclonal variation to occur in the calli. So how do you detect somaclonal variants? You have many techniques. Morphologically, you can detect by, you can measure the height, flowering date, leaf size. Definitely there will be variations in the qualitative characters. Quantitative is yield of flower, seeds, you know whatever is phenotypically expressed qualitatively as well as quantitatively, you can use for detecting variations. Okay, so analysis of morphological characters is the only thing which is used by the farmers, because we cannot go in for what to say, cytological studies or detection, DNA detection studies la nama, we cannot do it. So whatever is feasible in the field is analysis of morphological characters. So by seeing this, they can definitely be sure that they are, if they are a product of tissue culture, definitely it is a somaclonal variation. So they can use cytological studies. You can stain the nucleus you the root tip and leaf, you are doing anther pollen, what to say, you are doing the stratoscantia anther meiosis staining பண்றீங்கல்ல, அதே மாதிரி they will use the root tip and leaf tip. They'll stain it with astrocarmine and look for the changes in the chromosome. Either the morphology of the chromosome will show some change, or the number of chromosomes will be different. So this is very easy to perform in a lab. In a small lab you can do. Variant detection is you have to make use of cytophotometer, by which the nucleus can be stained to measure the amount of DNA. So looking at the difference in the amount of DNA, you can detect the somaclonal variations. Otherwise, you can use gel electrophoresis to see the differences in the amount of protein. When you target a particular protein, you want to see how is there a variation in the different generation of the callus. You can look for the concentration of the enzyme, concentration of a particular protein, concentration of pigments. You can look for specific secondary metabolites. You can detect them using gel electrophoresis. You can also look for resistance. For example, if you are raising a resistant variety against a particular chemical, you can look for whether the particular callus is definitely resistant to the chemical. You can look for you can include the toxin at a particular concentration in the medium and look for the growth of the callus. If the callus is effectively resistant to the toxin, it will grow. So that way you can detect for disease resistant variant or herbicide resistant variant. Okay, so in this case the plantlets will be the culture medium in which the plantlets are growing will be added with the herbicide, with the proper appropriate concentration of the herbicide. And so by this they will show that this particular plantlet is resistant to herbicide. Herbicide resistance is not a common feature, it is an acquired feature. It has to be acquired. So somaclonal variation is a very common acquiring, it helps in acquiring the resistance capabilities. So otherwise you can also detect environmental stress tolerant variety, like for example high salt variety rice, like in drought conditions, in drought areas where there is a saline soil, in areas where you have large area of saline soil, in that case you have a certain variety of rice which can grow well in saline soil. So if you can raise a rice variety over tissue culture to be resistant to a particular concentration of saline, you can raise that rice in the soil, particular soil. So selection of waterlogging and drought resistant tomatoes have been produced. Selection of temperature stress tolerant pears have been produced. Selection of mineral toxicity tolerant in sorghum, aluminum toxicity. So wherever you have places which raise a lot of sorghum, which cultivate a lot of sorghum, and which have aluminum ore, they have the mineral aluminum ore in the soil, in that case they can be at an advantage to raise such mineral toxicity tolerant varieties. So the scientists will be doing various changes, environmental stress they'll give to the cell lines, and they will release somaclonal variants as per the need for the farmers, as per the need of the area of cultivation, okay, and the quality of the soil. So that such somaclonal variants will be released periodically by the agriculture universities. So these are the factors which affect variation: selection, the protoplasm, which is the explant selection, what you want it to be resistant to. You want to develop a somaclonal variant against a toxin or a herbicide, or what is the agent that should be selected. And then the technique is also important, how you go about producing the variant, how long is the culture, the time duration of the culture, definitely the hormonal effects, and how stable is the resistant substance. That also plays a role in the development of the variant. Okay, so and then testing procedure, the process of testing, And regeneration capacity. The percentage of regeneration is also important. That affects the variation. So the variation should not be lost in few generations itself. It should be persisting. And the genotype and the explant source is also important. So explant should be a strong, it should have a strong genotype. It should be actually, what to say, it should be less prone to mutation. It should not be a, you know, more prone to mutation. அந்த மாதிரி ஒரு explant இருக்கக்கூடாது. You should have a very stable explant. That means you will get the explant from a plant which is grown in the most normal conditions, most favorable condition. It should be growing in a very favorable condition. Such plant length, if you take explant, the explant will have a very stable genotype. So stressed condition lende explant எடுக்கக்கூடாது. You should always take the explant from a well grown plant, grown in a very favorable environment. So in that case, the genotype of the explant is also very strong. So you can isolate somaclonal variants. You have a variety of cell callus culture. How are you going to isolate them? How are you going to find out, isolate somaclonal variants from a group of calli? So you can use without in vitro technique and with in vitro technique. You have two techniques. Without in vitro technique is that you use the callus, unorganized callus grown in the cultures that contains no selective agents. Okay, so they are first developed into the whole plant, and then you allow the callus to you add the particular chemical or resistant for which you are developing resistant into the medium. So the explant is cultured on a suitable medium supplemented with growth regulators, and the callus and cells do not contain any selective agent, that is toxic or inhibitory substance. These cultures are normally subcultured, transferred to shoot induction for regeneration. Now this produced plants are grown in pots, transferred to field and analyzed for somaclonal variants. So actually you don't induce or introduce any. Suppose for example you are producing a callus, you want to produce a tissue culture micropropagated plantlet which is resistant to a particular herbicide 2,4-D க்கு resistant இருக்கு. You need a plantlet developed from a callus. So in that case what they'll do is they will grow the callus in the most favorable conditions, and then directly after the regeneration is over, after you got the regenerated plantlets, then they will test for the herbicide resistant. So that is the without in vitro technique. With in vitro technique, cell lines are screened from cultures by their ability to survive in the presence of a substance in the medium. So with in vitro technique, in the medium itself you add the substance. Okay, so in lower concentrations you add, and then you slowly increase the concentration and you get a culture line or the cell line which is resistant to the particular chemical. So the differentiated callus obtained from an explant is exposed in the medium to the inhibitors. So with in vitro technique you have a process in which you add the particular inhibitory agent into the medium. So the callus, growing callus is exposed to the inhibitory substance right from the start. Okay, so the plants are in vitro screened against the toxin. So if it grows, that means it is resistant. If it is able to grow and develop into callus, they are able to develop shoot tips and root tips, and they are able to develop into plantlets with the inhibitory substance in the medium. That means they are resistant to the chemical. So subsequent generation, the plants resistant to the toxin are selected and grown further by vegetative propagation or self-pollination, and then after many generations only it is given to the field. Okay, so subsequent generations are analyzed for disease resistant plant against a particular specific pathogenic organism. So this in vitro technique, with in vitro technique is more stable and more reliable than without in vitro technique. In this case you are not you're not adding the particular chemical. You are only growing the callus in normal conditions, favorable conditions, and then after the plantlets are regenerated, then you are exposing them to the chemical. So this in vitro technique, the callus, when it is in the growing stage itself, it is exposed to the chemical. So this is more reliable technique. So the specific selection for isolating a desirable trait you can use in vitro technique, and it is less time consuming because throughout the micropropagation process, you are able to see which callus is surviving. In the previous technique, you don't have any method to see whether the callus is surviving or not, because you don't include any chemical into the medium. Only in the last phase, after the regenerated plantlets are produced, then you are going to test whether it is resistant or sensitive. But in this case, during the whole process itself, you are able to find out whether the callus, which batch of callus is resistant. You can detect it then and there. So the process is less time consuming as compared to the without in vitro approach. So callus tissue is taken after organogenesis, regenerated plants, hardening and selfing, somaclonal variation. You get what is called the somaclonal variation. So this is the flow chart. You take the explant, develop the callus, maintain the callus. Okay, small pieces of calli in the medium with the toxin. Okay, or it can be a pathogenic organism. You are going to develop a, plantlet which is resistant to a pathogen. Na? You use the pathogenic organism, culture it, purify the filtrate, isolate the toxin, and then determine the lethal concentration, add it in the medium. And then you have to go grow it in different cycles, selection cycles, different generations, isolate the tolerant calli, and then regenerate, develop into a plantlet. Again, in vivo screening. You have to do in vivo screening. You have to develop subsequent generations. This is the R naught. This plantlet is the R naught. This is the R naught. So you have to develop generations R1, R2, R3. You have to develop subsequent generations from R naught and then given for agronomical trials. Then you have to give it to the field for trial cultivation. So this is the flow chart for detection of somaclonal variation. So applications, definitely they are very important agronomically useful crops. Select, you have different variations which are selected based on the crop, and resistance to diseases. Somaclonal variations have been very important in developing crop variants which are resistant to diseases. Stress, abiotic stresses like freezing tolerant wheat have been developed, salt resistant rice has been developed, aluminum tolerant vegetables, no, carrot, sorghum, tomato have been developed. Again, herbicide resistant plants have been developed, and seed quality improvement is seen in using somaclonal variation. So you can create additional genetic variations whatever you want. You can introduce variation and create a variant, and you can increase the production of secondary metabolites in calli by inducing somaclonal variations, and then suitable for breeding of tree species. The somaclonal variants are suitable for breeding for tree species. There are some disadvantages. So you cannot expect uniformity. You cannot expect, because it is it is as it comes, it can even go predictable kadayadu. Okay, fine, you are having a particular protocol by which if you use that protocol you will get stable changes, stable variation you have. But again, it is not predictable in the field condition. It may be predictable in the lab conditions, but it may not be predictable in the field conditions. So you cannot say that you will get uniformity, or where tissue culture is employed for rapid propagation. So in horticultural area, you cannot say that clonal variation, somaclonal variation will be uniform throughout. Suppose you are distributing 1,000 plantlets, you cannot give a guarantee that all the 1,000 plantlets will be holding the variant with them. Okay, that is the condition. It sometimes lead to undesirable results because of mutations. Somaclonal variation in the genes will also have higher mutation rate. So you can always have undesirable results, and selected variants are random and genetically unstable. That is what I have been telling before. And you require extensive field trials. You just can't try once and then see that, yeah, I have generated. Subsequent generations may lose the variation also, and not suitable for yield and quality, definitely, because yield is a very unstable. Somaclonal variation la quantitative variation is very very unpredictable. Yield ஒரு generationல இருக்கும், அடுத்த generationல it might not be there because it is a somatic variation. So it is not a true genetic variation, so it is very unpredictable. And then some may develop variants with pleiotropic effects. Some may, batch வந்து மாறுதல் இருக்கலாம். ஒரே வேரியேஷன் வந்து it might change in the same field. You might have 1,000 plants with one variation and 1,000 plants with another variation. That's why they say pleiotropic effects will be there in variants. So gametoclonal variation is the same as somaclonal. The only thing is haploid plants regenerated from callus culture which show genetic variation is called gametoclonal variation. So such variation may be subjected to genetic because they will be more stable. They will be more stable, and because all the cells will be haploid, all the calli or whatever the plant regenerated from gametes will be haploid. So they will be more stable also. So the variation is more desirable. You can also make it diploid. So even though the cells are haploid, you can use chemical treatment and make it diploid so that the variation will be even more enhanced. So yield in pathina, suppose the size of the mango is a you are getting it as a gametoclonal variation. You can enhance the size of the mango if you can change the ploidy of the cell callus. If the cells are all haploid, if you can make it as diploid, you can get a better yield. So gametoclonal variations are more stable than somatoclonal variations. So this is the difference between somatoclonal variation and gametoclonal variation. So it involves fusion of somatic cells. In this, gametoclonal variation involves fusion of gametes. So you can produce diploid plant from gameto, because normally it produces a diploid plant. But in somaclonal, it involves the production of allotetraploid plants. You can get different ploid plants. There is in somaclonal variation, there is equal contribution of cytoplasm. See, it is going to stop now. I think you can rejoin, please.
Okay, ma'am.